Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa, where I take difficult biological concepts and make them easy to understand. In our last video, we looked at the different types of muscle tissue and what makes each unique. Today, we're zooming in on skeletal muscle and exploring its structure from the outside in. Skeletal muscle may seem simple, it contracts to move your bones. But when we peel back the layers, there's an incredible organization that makes this possible. We'll look at the connective tissue layers that surround muscle, then we'll look into the microscopic anatomy of individual muscle fibers. Skeletal muscle is highly organized. It's not just a bundle of random fibers, it's arranged in layers, each wrapped in a connective tissue for support, protection, and force transmission. Let's start looking at the different connective tissue layers by looking at the outermost layer. The epimysium is a fibrous sheath of dense irregular connective tissue. This is the outermost covering that wraps the entire muscle. This layer protects the muscle and gives it shape. It connects to tendons, which anchor muscle to bone. As we move in further, the perimysium is a layer of thick, dense, irregular connective tissue that wraps groups of 10 to 100 or more muscle fibers, separating them into bundles called fascicles. Fascicles are visible to the naked eye. You've seen them in a cut of meat when you look at that grain of the meat. This organizes the fibers so that they can contract in unison. And as we move even deeper, we reach the endomysium. This is the innermost layer that surrounds each individual muscle fiber or muscle cell. This layer also provides electrical insulation and houses capillaries and nerves. The connective tissues not only protect and organize, they also extend beyond the muscle fibers to merge into a rope-like tendon that attaches the muscle to bone. With the tendon attached to bone, when a muscle contraction occurs, we can then have the movement of that bone. The other type of connective tissue that we see in muscle is called the fascia. This is a sheet of dense connective tissue that supports and surrounds muscles and other organs of the body. This holds together muscles with similar functions, and we see it again surrounding the entire muscle. The fascia also allows free movements of muscles, carries nerves, blood vessels, and lymphatic vessels, and it fills the spaces between muscles. Now that you understand a bit about these connective tissues that are surrounding these different bundles of muscles to protect it, provide blood vessels and nerves, that nourishment so that it can survive and contract at the same time, let's move down to the microscopic level and look at a muscle fiber. Remember, in the last video, we went over skeletal muscle cells. So a muscle fiber is a single muscle cell. They are very, very long, cylindrical, and multinucleated. This is because during development, little cells called myoblasts fuse together to form these big, strong fibers. Once that fusion happens, the cell loses its ability to undergo cell division. So you're basically born with all the skeletal muscle fibers you'll ever get. So now that we know a muscle fiber is just a really large multinucleated cell, let's take a closer look inside. The first structure we want to notice is the sarcolemma. This is the muscle cell's plasma membrane. Like any cell membrane, it keeps the intracellular environment and the extracellular space separate. But in muscle cells, it has an additional special role, conducting electrical signals. These impulses, or action potentials, travel along the sarcolemma to initiate the process of contraction. But the signal can't just stay on the surface. It needs to reach deep inside the fiber. That's where T-tubules or transverse tubules come in. These structures serve as passageways that carry the action potential deep into the muscle fiber. This makes sure that every part of the muscle cell gets the message quickly and uniformly so that it all contracts at the same time. Next, filling the inside of the fiber, we find the sarcoplasm, which is the muscle cell's cytoplasm. Beyond containing the usual organelles, the sarcoplasm is enriched with glycogen granules, which provide a ready source of glucose for energy, and myoglobin, which is an oxygen-binding protein that supports aerobic metabolism. Together, these reserves allow the muscle fiber to keep working. The most prominent structures within the sarcoplasm are the myofibrils, cylindrical, thread-like arrays that fill most of the sarcoplasm. Myofibrils are composed of repeating contractile units called sarcomeres, which are the contractile units of the muscle. We'll get into sarcomeres in more detail later, but for now, just remember myofibrils are what gives skeletal muscle its striated appearance and its ability to contract. Surrounding each myofibril is a specialized form of the smooth endoplasmic reticulum called the sarcoplasmic reticulum. Its main job is to store and release calcium ions, which are essential regulators of contraction. When an action potential travels down the T-tubules, it triggers the sarcoplasmic reticulum to release calcium into the sarcoplasm. This rapid change in calcium concentration is the signal that activates the myofibrils to contract. So within each muscle fiber, the sarcolemma conducts the electrical impulse, the T-tubules distribute it into the fiber's depth to stimulate the sarcoplasmic reticulum, which then releases calcium, and the myofibrils carry out the contraction. But to really understand contraction, we need to zoom in one step further to the sarcomere. Now that we've covered the major structures of the muscle fiber, let's go in further to the myofibrils and see how contraction actually happens. Each myofibril is organized into repeating units called sarcomeres, which, like I mentioned before, are the basic contractile units of skeletal muscle. Sarcomeres are what give muscle its striated or striped appearance underneath the microscope. A sarcomere is defined by its boundaries, the Z-discs. It runs from Z-disc to Z-disc. These discs anchor the thin filaments, which are made mostly of actin. These thin filaments, or actin, extend from the Z-disc toward the center, while the thick filaments, composed mainly of myosin, sit in the middle. The overlap between the thick myosin and thin actin filaments is where the action of contraction takes place. We can actually see these arrangements as light and dark bands. The I band is the lighter region, containing only thin filaments. The A band is darker, spanning the length of the thick filaments, even where they overlap with thin filaments. Within the darker A band, there's a slightly lighter region in the center called the H zone. This area contains only thick filaments, no overlap with thin filaments. Right in the middle of the H zone is the M line, which anchors the thick filaments in place. Here's what happens during contraction. The thin filaments slide inward toward the M line, pulling the Z-discs closer together. As this happens, the thin filaments, actin, are sliding past the thick filaments, myosin, shortening the sarcomere. The I band and H zone narrow, and in some cases, the H zone can even disappear. The A band, however, remains the same length because the thick filaments themselves do not shorten. This is called the sliding filament model, since the actin and myosin slide past each other to shorten the sarcomere and contract the muscle. We will be looking at this in much more detail in the next video. Today, we broke down skeletal muscle from the outside in, looking at the connective tissue layers, muscle fibers, and finally the sarcomere. This incredible organization allows muscles to contract with precision and power. In our next video, we'll build on this by exploring how muscles actually contract, looking at the sliding filament mechanism in action. If you enjoyed the content of this video, please make sure to subscribe to this channel, give this video a like, and turn on notifications so that you never miss out on new content. Thanks for watching, and I'll see you in the next video.